Hello students, welcome to this tutorial, basically physiology of blood. So in the physiology of blood, we are going to focus on part 2, which is specifically the formed elements. Under formed elements, we are going to talk about two major formed elements, that is the white blood cells, also known as leukocytes, and then second, we're also going to look at the platelets as well, looking at the blood clotting mechanism. So let's get started. So now our today's objectives are that at the end of this session, students should be able to understand the structure and the function of leukocytes and the immune responses. Okay, so we need to understand the structure of the white blood cells, and then we also need to understand the structure and functions of platelets and the hemostasis. So basically platelets, this is a formed element that help in hemostasis. So helping platelets, understanding platelets is going to help us understand how blood clotting mechanism comes about. Yes. So now, like I mentioned, we are first going to start with the white blood cells, the leukocytes, with their immune response. So we know that the white blood cells plays a critical role in the body, and that is to defend the body against invading pathogens. So we basically want to look at the structure of the white blood cells, possible classifications, and what each and every classification do. Okay, as far as immune response is concerned. So now we are going to look at different types of white blood cells. Basically, the white blood cells are in different types. So we have what we call, these two types are categorized as the granulocytes and the agranulocyte. So now, these are the two major classifications of white blood cells, the granulocytes and agranulocytes. When we say granulocytes, what are we basically looking at? The granulocytes contain granules, and these granules could be basic or acidic. Okay. However, agranulocytes do not have granules in their nucleus. Okay. So what do I mean? We have 5 types of white blood cells, and then let's basically look at those particular types. So under these 5 types of white blood cells, we have eosinophil, we have the basophil, and we have the neutrophil. These 3 types are called granulocytes. Why? Because they have granules. As you can see, when you look closely inside there, you can see some kind of granules. These granules are acidic and basic combination. Yeah, we're going to read more about them. Then we have 2, the monocyte and the lymphocytes. So the best way to know this, just know that all the cytes are granulocyte, meaning they do not have granules in their nucleus. So when you look at the monocyte, it's ending with cyte, and then lymphocyte is ending with cyte. So these two are called agranulocyte because they don't have granules in their nucleus. I hope you are following through. So now, let's try to look into the properties of white blood cells. What I was mentioning, we're also going to look at them in more detail. So now, when we look at the white blood cells are described to be the only formed elements that are complete cells. When you look at the formed elements, we have the red blood cells, white blood cells, and platelets. The red blood cells as well as platelets, these are incomplete cells. Why? Because they lack some important cell organelles for their survival. But these white blood cells, they have all organelles, the nucleus, the mitochondria, the cell membrane, everything that this particular cell needs to survive. Hence, we are going to call them complete cells. I hope that makes sense. Then the question is going to be asked there, why are white blood cells called complete cells? It's just because they have all organelles that can sustain them. Mitochondria, nucleus, I'll repeat again. Okay, so now, under normal circumstance, the normal number of white blood cells in the body should be in between 4,000 to 10,000 per millimeters cubed of blood. So when we look at this, every millimeter cubed of particular blood should contain 4,000 to 10,000 of the white blood cells. This range is coming in, it depends. Under normal circumstances, when there is no infection in the body, 4,000, 5,000 is normal. And in some cases whereby someone, the body has some infection, these white blood cells increases in number so that they can fight that particular infection. So this can even go up to 10,000 millimeters cubed, so that we can help combat this particular infection that has arrived. Okay, then apart from that, so these are very much important points when we are talking about the concept of full blood count. When we're talking about full blood count, it's an investigation that looks at the blood profile. So this particular blood profile, we look at all the components: the hematocrit, the red blood cells count, the hemoglobin count, white blood cell count, everything else, all the counts we put them together so that we see if at all the blood profile is okay. That's what we mean to say FBC, full blood count. Even white blood cell number is need to be taken. So it's very much important for you to know the normal number of white blood cells in the body. Then apart from that, we also have another property of white blood cells. So basically, the next one is mobility. What is about mobility? Look, these white blood cells are deemed to defend the body against pathogens. So they have a very unique property that enables them to fight this particular infection that is invading the body, and that one is mobility. How about mobility? The white blood cells are described to be highly mobile and rich tissue fluids. So the white blood cells are able to move from one place to another, okay, for as long as that particular area has been infected. So there's a particular way on how these particular white blood cells move, and we're going to look at it as we go on. So now, the thing is that the other property of white blood cells, like I was explaining, to say that range that has been given on top there differs. Under normal circumstances, when there is no infection, white blood cell number just remains like that. But then the body has put itself in a condition to say, in case where there is high infection, the white blood cell number need to multiply so that they can combat that particular infection. So this is now the property that we are looking at, to say when infection occur, the white blood cells increase in number. Specifically, when you have neutrophils, most of the time these questions are going to be asked in MCQs. How are these questions going to be asked? They are going to tell you, during infection, which of the following type of white blood cells is going to be more, is going to be observed, or is going to have multiple numbers or increase in number. So that particular answer is going to be neutrophils. So when an infection occurs, the white blood cells number increase. Specifically, when we are looking at the neutrophils increases in number when an infection has occurred. Then it's very much important that we know about the white blood cells. Okay, the other property about white blood cells is the lifespan of white blood cells. So you are going to find that most of the white blood cells, we can say many, okay, they live only a few days. So now, they live only a few days. This may be due to their engagement with pathogens, okay? Because the white blood cell, it's more like the soldier, okay? I'm going to give an example of an officer who is working from their office, and then we have a soldier who has gone for peacekeeping. Their life is more at danger than an officer who is an office, just operating on computers and everything. You understand? So basically, that's what we mean to say. Because white blood cells interact mostly with pathogens, these pathogens have chemicals that destroy these white blood cells, and that is the reason why white blood cells, they die within a few days. Okay, so now we must be able to note that the white blood cells are categorized into two main categories. Now let's look at these two main categories I earlier mentioned. These two main categories, we have granulocytes. Okay, granulocytes. So how about the granulocyte? These granulocytes we have here on top, we have neutrophils, eosinophil, and basophil. They contain specialized membrane-bound cytoplasmic granules. Okay, in their cytoplasm, they have stains. Specifically, we have acidic, basic, or that particular neutrophil or that particular white blood cells can have both. Okay, so now these granules are what differentiates these three from the other two that I mentioned. I said the other ones end with site. All the two sites, monocytes, lymphocytes, those ones they are granulocytes because they don't have granules. And with the granulocytes, they are actually larger. Okay, these granulocytes, they are larger and they can live longer than the red blood cells. We all know the lifespan of red blood cells. Within 120 days, the red blood cells dies. So in as much as we say that the white blood cells dies within a few days, but I mentioned to say not all of them, but most of them. Now, amongst that few number that survive for a long period of time, we have granulocytes. Okay, then one thing about these granulocytes is that they have lobed nucleus and they are all phagocytic cells. So what do we mean when we say they are all phagocytic cells? They are able to ingest a particular pathogen that is coming. You understand? So they are able to phagocytize a certain foreign substance that is coming. This is what we mean, they are phagocytic cells. So now, finally, the granulocytes include the neutrophils, the eosinophils, and the basophils. Now, let's look at the granulocyte. So a granulocyte, we are saying these ones, they lack obvious granules. Granules, they are those stains that were inside. You can see there are these stains inside. You can see them. But when you look at these, there are no stains inside. And these two types of white blood cells, we have monocyte and the lymphocyte. They are all sites. In case you have issues with memorizing the one or knowing them, just know that they end with sites. Everything that, all the two that ends with site from the group of 5, then just know that they are a granulocyte. So now let's jump into more detail of each and every white blood cell so that we know their function and they are also individual properties, because they are also very much important. Most of the time MCQs in physiology are going to be asked depending on individual differentials. Like we want you to understand what differentiates a neutrophil from eosinophil, what differentiates basophil from monocyte, what differentiates a monocyte from lymphocyte. That is what physiology is going to require you. Even when you are studying, you should be able to know the differences. In what condition are the basophils the most? How are you going to differentiate eosinophil from neutrophils? That is what we are basically looking. And then apart from that, we also want to look at the prevalence. Amongst these 5 white blood cells, which ones are the most common, or which ones are higher in number, like total number of white blood cells in the body? So that is the angle questions are going to come in physiology. So now let's try to explore more about what I've said. So now let's start with the granulocytes. We know when I talk about granulocytes, we know now that these are particularly having some stains. These stains can either be acid alone, can either be basic, or both of them can be in this particular white blood cell. Now let's look at the neutrophil. The neutrophil, it is a granulocyte and it has stains. Okay? What is unique about neutrophil is that the neutrophil, they are the most, like they're the largest number of white blood cells in the body. They account to about 60 to 70% of the entire total of the white blood cells. They are neutrophils. These now are the most important MCQ questions in physiology. Okay. Then the diameter of these neutrophils is about 10 to 15 micrometer. So basically, why is it important for you to know the diameter? Okay, questions are going to arise to say, something that is large inside, like they are large, that's why they are phagocytic. So they are large, that's why they can ingest. When we're looking at phagocytic cell, it's a cell that is able to ingest something, just like the way the human mouth is. Okay, the only thing that you can accommodate in your mouth is when it fits the size of the mouth. So these ones are large enough to ingest the pathogens. And what is unique about the neutrophils? Please take note of this. Even in pathology at third year level, when you start learning inflammation. So basically, the inflammation process, we say the first type of white blood cell to arrive at the site of infection are the neutrophils. These are the first one to arrive at the site of infection. So now, when you look at the nucleus, it's when you look at the nucleus, the neutrophils usually have about 2 to 5 lobes. Okay, as you can see here, when you count them, 1, 2, 3, 4. This is the nucleus of the neutrophil. So they have about 2 to 5 lobes. Okay, and they increase depending on the age of the cell. When do the neutrophils come to the site of infection? Or when do the neutrophils called upon? When do they arrive? Or what makes them to be readily available in the body? We are talking about in case of stress, in case of burns and bacterial infection, the neutrophils are always there. Okay? Then it decreases in case of radiation exposure and when B12, vitamin B12 deficiency. This decreases the neutrophils. Then let's go to eosinophils. The eosinophils, they account for 1 to 4% of the total white blood cells, and their size is 10 to 12 micrometer in diameter. They are about 2 to 3 lobed. The neutrophil had about 2 to 5. This one is 2 to 3. Then they increase in case of allergic reactions, then parasitic infection, and autoimmune disease. That's when the eosinophils increases. So they are able to kill parasitic worms, destroy antigen-antibody complexes. When we say antigen antibody complex, we mean to say the antigen, the worst is the antibodies are there to protect the body against infection or foreign substance. So when we say antigen-antibody complex, meaning that the antibodies have found that particular antigen that is a problem to the body. So these eosinophils come to destroy them, meaning the antibodies are the marker for the destruction of this particular complex. Then they inactivate some inflammatory chemical of allergy, for example histamine. So in case of allergic reaction, eosinophils is going to come and inactivate histamine so that some infection can be reduced. For example, some people when they have allergic reaction, you are going to see that they develop rash and everything. So that's what we basically want to prevent, because those particular things, like, it will become itchy and everything. So we want to avoid that itching, and that's why we are relieving symptoms. Then how about basophil? Basophil accounts for 0.5 to 1% of the total white blood cells, and it liberates heparin and histamine during allergic reaction. Okay? Then it intensifies inflammatory response, the basophil. So now, when does basophil get to increase in the body? In case of allergic reactions, leukemia, cancers, hypothyroidism, these conditions make basophil to increase in the body. Then they also decrease during pregnancy, ovulation, stress, and hyperthyroidism. Then now let's look at the agranulocytes now, the monocyte and the lymphocyte. When you look at the lymphocytes, they make up 20 to 25% of the entire total white blood cells. Okay? So they are usually small because their diameter is about 6 to 9 micrometer. Okay? Then when it is large, then it's about 10 to 14 micrometer. The nucleus is usually round or slightly indented. Okay? So what is one thing about lymphocyte is that the lymphocytes are the ones which produces what we call the areas of antibody production. You've heard of antibodies. What? Lymphocytes are the ones which produces antibodies. Okay? The precursor cells being the plasma cell, then the plasma cell produces the antibodies. B cells produces plasma cells, and then plasma cells produces the antibodies. Okay? Then they also, the lymphocytes, because we have two types of lymphocytes. We have the B cells and the T. So we have B lymphocytes and T lymphocytes. The T lymphocytes, they attack viruses, cancer cells, and transplanted tissues. Then under the lymphocytes also have natural killer cells. They attack infectious microbes and tumor cells. So when you look at B cells, T cells, and natural killer cells, they are part of the lymphocytes. And we basically mean to say B lymphocytes, T lymphocytes. B lymphocytes is where the concept of antibodies come to play. But in case of T cells, that's where now we talk about CD4 cells and CD8 cells. The CD4 cells, they are very much important and they are major target for HIV virus. Okay? They are helper cells. And then we have the CD8 cells. These are cytotoxic cells, like they kill cancer cells just like that. Then finally we have monocytes. They account for 4 to 8% of entire white blood cells, and they are the largest white blood cells. Monocytes are the largest white blood cells. So this, you should be noting this. We've been talking about white blood cells in terms of number and in terms of size. So we're saying in terms of number, the highest number is the neutrophils, because it's about 60 to 70% of the total white blood cells. However, when I look at the size, the monocytes are the largest ones. Okay? So the monocytes, these ones, in other words, we can call them to say that monocytes are macrophages. Why? Because they are more like precursor cells. When we say precursor cells, we are meaning it's a parenchyma where some cells are going to originate from. So they leave the circulation, which is the bloodstream, and enter tissue, and they are going to differentiate. Differentiate is more like changing the form from monocyte to macrophages. So when you hear the word monocyte, we are literally talking about macrophages in tissues. You understand? So what happens is that the macrophages activate lymphocytes to mount an immune response. So once the lymphocytes are activated, they produce those cells I was telling you, the B cells and T cells. The B cells produces antibodies. Always know. Questions are going to arise. What is the purpose of B cells? They produce antibodies. How about T cells? T cells, they are cytotoxic killer cells. Why? Because these are CD4 cells and CD8 cells. They are there to kill the, let's say, cancers, everything. Okay? Then they phagocytize bacteria, dead cells, and other debris. And this is the function of macrophage. Macrophage is just to make sure that they remove any worn out tissue. Okay, this is the reason why some kind of autoimmune diseases, happen is that when the destruction of these formed elements occurs, like the red blood cells, when red blood cells are destroyed, they are going to be marked for destruction. Okay, And when it's a condition that is leading to this destruction of red blood cells, then there's going to be severe, too much destruction of this particular cells. So that too much can lead to hyperactivation of the immune cell. When too much of the red blood cells are like they are marked damage, then the immune system can be hyperactive. Okay. So now, how does these are some of the questions that you can be asked, like how does the white blood cells move through the tissue? Remember, we talked about neutrophils being the first white blood cells to reach the site of infection. Note that. Okay, you need to note that down. So how do they move from the bloodstream for them to go to the tissue? Because from the bloodstream, remember, under physiology, specifically introduction, we talked about two major fluid compartments. We have extracellular and intracellular. So when you're talking about plasma is an extracellular fluid, and for it to reach the tissues, it needs to cross the plasma capillary, okay, the plasma membrane, capillary membrane, so that it can find itself in the interstitial. You should review the physiology of the introduction to physiology in case you've missed out some information. Kindly request it in the comment section. Introduction to physiology tutorial. So basically, these white blood cells need to leave the bloodstream and go to the tissue, but they need to cross the capillary membrane. How do they cross the capillary membrane? These are some of the ways on how a white blood cell is going to leave the bloodstream and find itself in the tissue. The first one is what we call diapedesis. What is diapedesis? Diapedesis is when the white blood cells squeeze out through the capillary pore, because we say the capillary membrane becomes leaky in terms of inflammation. When an infection has arrived, there's what we call activation of inflammatory mediators like histamine, bradykinin. These particular mediators comes in so that they can make the capillary membrane porous so that they can allow these particular white blood cells to move through. So in this case, we have neutrophils and monocytes. These ones are able to squeeze out through the capillary pore, and we call that process diapedesis. Okay, then the second one is amoeboid motion. How is amoeboid motion done? These white blood cells, they produce something what we call pseudopodia. The pseudopodia is more like, pseudo means force, okay? Pseudo means force. So pseudopodia, we mean they produce force legs, okay? These legs are going to be produced, they are force, and they are going to reach the microbes in the tissue. And then we also have what we call chemotaxis. The chemotaxis, this is just from the word itself. The white blood cells utilizes some chemical mediators that are present there, so that these particular white blood cells can locate where exactly the infection is located. So the white blood cells are attracted by chemicals or toxins produced by the microbe or inflamed, so that they can reach the site of infection. Okay? Then we also have what we call phagocytosis. The phagocytosis is just the engulfing and destroying of a particular pathogen. For example, this happens with neutrophils and macrophage, specifically macrophage. I've told you to say macrophage is just a unique name of a monocyte when it has reached the tissue, it has crossed the capillary membrane. Okay, so these are some of the mobility through the tissues, how the white blood cells manage to go through the tissues. You know, there's a biological barrier, and that is a capillary membrane. They need to cross it so they can find themselves in the interstitium. Interstitium, that's where we're talking about the tissues. I hope we're together. So these are different processes. For example, chemotaxic source. If this particular white blood cell is moving from this blood, and the kind of infection is here, this particular antigen is going to release some toxins. So the body uses different mediators that are going to help this particular white blood cell to reach this area of infection. So whenever there's an infection, there are some things that favors the movement of these white blood cells. For example, we have increased permeability, which is capillary permeability, so that white blood cell can easily move. You can see them moving towards the source of these particular microbes. Okay, so this is the epidesis movement. This is the process of the epidesis that we were looking at, whereby they just squeeze through because the pores are leaky. They have become leaky, and then they squeeze through to reach the site of infection. Okay, so some of the clinical correlation to this topic is that we have some conditions like leukemia. What is with leukemia? Leukemia is an increased white blood cell count, white blood cell number. Remember we said the white blood cell number is about 4,000 to 10,000 millimeter cubed. Okay, so we mean to say per millimeter cubed of this particular blood, we need to have about 4,000. I said this range is there because there are some instances where the white blood cells are not needed in the body. That's when they are in average, like 4,500. But in case of infection, they can go up to 10,000. Going above 10,000 millimeter cubed, it means that there is probably an autoimmune disease. Okay, when we say autoimmune disease, we mean to say the body has failed to recognize its own tissue and is destroying itself. We've seen that in conditions like diabetes mellitus. In diabetes mellitus type 1. Diabetes mellitus type 1, that's an autoimmune disease whereby the beta cells of Langerhans in the pancreas get to be destroyed by its own immune system. Okay, so now the cancerous, usually in leukemia, there is cancerous production of white blood cells. They increase in number. Okay, these occur in the body blood forming tissues. It happens in the bone marrow and it happens in the lymphatic system. These are what we call body's blood forming tissue. They are the ones responsible for forming blood. So they are going to be corrupted so that they can produce duplicates. These duplicates, they are not original of the white blood cells. Hence the number increasing from where above 10,000. Then their increased production takes the space of platelets and red blood cell count, causing anemia. So in leukemia, someone can present with anemia because we have a recommended number of white blood cells and red blood cells as well as platelets. The increased production of the other formed element can lead to the increment of another. Okay, I mean can lead to the decrease in number of the other cells. So this is what happens in leukemia. You can see that someone has anemia because the space for the red blood cells has been taken up by the white blood cells. This brings into anemia. So that's it. And then we also have what we call leukopenia. When I look at leukopenia, this is decreased production of white blood cells. Usually happens when the bone marrow stops producing them. Okay, remember we looked at hemopoiesis. This is a process we said, this is a process by which all cell lineage get to be produced. So if the bone marrow, specifically the red bone marrow, is responsible for production of all cell lineage, if it stops producing cells, then it's going to happen. Usually this happens in drug poison or kind of X-ray, especially repeated X-rays or unprotected ones. Then someone is more likely to have leukopenia. All right, so basically these are some of the practical hematology or leukocyte differential. So when we mean to say this, we are saying in case someone has gone for a full blood count and then they see that there is an increased production of neutrophils, increased production of lymphocytes, increased production of monocytes, increased production of basophils, increased production of eosinophils. How are you going to know that increased production of neutrophils is going to lead to this particular infection? So this is these are the differentials, how you can differentiate one disease from another. So if there is a high increment in the neutrophils, just know that suspected is bacterial infection. Not even suspected, just know that this is a bacterial infection. Okay, in case someone presented with some signs and symptoms of an infection, and then you request full blood count, and then the blood count comes with a high number of neutrophils, then just know that that's a bacterial infection. So you are going to use this particular medication to treat that particular infection. Then if you have high number increment in the lymphocytes, just know that this particular is a viral infection. Okay, then if we have monocyte, just know that it's a chronic infection. Then if you see a lot of basophils, then just know that it's allergic reaction. Then if you see a lot of a high number of eosinophils, just know that it is a parasitic infection. And this is how you are going to differentiate the diseases using the white blood cell count of all its types of white blood cells. Okay, so basically now let's look at the platelets and hemostasis. When we look at platelets, we're actually even done with this. It's just few minutes, then we're done. Platelets and hemostasis. How about platelets and hemostasis? Platelets plays a critical role in blood clotting, and blood clotting stopping the blood. Hemo, blood, stasis, stoppage. So stopping blood, stopping hemorrhage. Okay, so what's with platelets and hemostasis? So what are platelets or thrombocytes? This is the other name. These are small non-nucleated, they are incomplete cells, round oval-shaped cell fragments. They are just fragments. Nothing really does make sense about them. Their size ranges 1 to 4 micrometer in diameter. As you can see, when you're looking at the white blood cells, they were about 6, 10 to 14 micrometer, but here we have 1 to 4 micrometer, meaning they are small fragments. Okay, then the cytoplasm is stained pale blue and contains many pink granules. They contain granules. They are produced in the bone marrow by fragments of megakaryocytes, which are large and multinucleated cells. Okay, megakaryocytes, these are what produces platelets. Let's know that particular point. So their primary function is to prevent blood loss from hemorrhage by forming a platelet plug. The normal value accounts to about 150,000 to 300,000 millimeter cubed of blood. What we mean is, if you get the millimeter cubed, 1 millimeter cubed of blood, you need to find 150 platelets. Then platelets have a lifespan of approximately 10 days. These questions, these are properties, and they are very much important questions that are going to arise from this. What's the lifespan of platelets? They are approximately about 10 days. Then synthetic platelets are removed by the spleen. Whenever a platelet is get to be damaged, they are removed by the spleen. Okay, the spleen. How are platelets attracted? Okay, these platelets are attracted to hemorrhage. Whenever someone is bleeding, platelets move closer to that area. Plaque leaks promotes constriction of blood vessels. Then they also trigger inflammation, and then they initiate clotting. This is what they do, because we say platelets are very much important for blood clotting, to stop blood from moving actually. So when plugs, then what happens is that the plug leaks, then promote constriction of the blood vessel. You know, constriction of blood vessel is going to narrow the like the amount at what volume is being how much you are losing this particular blood. So actually the constriction of blood vessels is going to help in the blood clot, and then they're also going to trigger some kind of inflammation. Okay, so during this particular inflammation, and then they are going to initiate the clotting using different clotting cascade. Okay, so now let's look at this same hemostasis. What is hemostasis? Hemostasis refers to the stoppage or arresting of bleeding. We want someone to stop bleeding. The process of the process of arresting bleeding is what we call hemostasis. The mechanism or action that limit or prevent blood loss They include blood vessel spasm, platelet plug formation, blood coagulation. So these are simple mechanisms that are involved in hemostasis. We are saying there is need to be a blood vessel spasm, then platelet plug formation, we call it primary hemostasis, then blood coagulation, we call this secondary hemostasis. Now, let's look at this table. What happens in blood vessel spasm? Get this. Questions are going to arise. What happens in blood vessel spasm? What is blood vessel spasm? Someone have had a cut, they have cut themselves. How is it going to happen? So imagine someone has cut themselves. They are having an open wound. What happens? Due to the direct stimulation to the vessel or wall to the pain receptor, platelets release serotonin, which is a vasoconstrictor. Okay? So it's going to release serotonin, which is a vasoconstrictor, because someone has been cut and there's direct stimulation to the vessel walls, and this is going to stimulate the pain receptors. Then what is the effect? Smooth muscles in the vessel wall contract reflexively, okay? Because there is pain, so the smooth muscles are going to contract reflexively. Then vasoconstriction helps maintain prolonged vasospasm, because the blood vessels have constricted and then the smooth muscles are contracting reflexively to prevent blood loss. Then what happens after that? We have what we call platelet plug formation. What happens? What is the stimulus for the platelet plug to be formed? Exposure of platelets to rough surface. When the platelets are exposed to the rough surface or to the collagen of connective tissue of the damaged tissue, what happens is that the platelet plug start to form. Okay? What is the effect? Platelets adheres to the rough surfaces and to each other, forming a plug. You are going to see this in the next slide. Then we have what we call blood coagulation. What is with blood coagulation? What's the stimulus? Cellular damage and blood contact with foreign surfaces activate factors that favor coagulation. Because there is a damage and then blood is in contact with foreign surfaces. Okay? Maybe it's at the wound of infection, the area of infection or the area of injury. So what happens is that blood clot forms as a result of series of reactions that we're going to look at, terminating in the conversion of fibrinogen into fibrin. We are going to look at this. What is this? Okay. So now let's look at the step by step. What happens is that whenever there is a cut into the wound, okay, whenever there is break into the vessel, what happens? The blood escapes through the break. Blood is going to be, it wants to leak. You can see the platelets. We are going to take those white grains to be the platelets. So these platelets are also going to come at the site of infection. So what happens here? You can see the number was small here, but look at here. The platelets adhere to each other. They adhere to each other and to the end of the broken vessel and to expose collagen. So they are trying to cover up this space. Then what happens in stage 4? Platelet plug helps control blood loss. The platelets have completely covered the area where blood was. This is the reason why whenever your blood profile is normal, the formation of plaque should be fast, and the healing and the hemostasis should be faster. Within few minutes it's done. So what is with blood coagulation? It's going to be triggered by cellular damage and blood contact with the foreign surfaces. Okay? A blood clot is going to form, like I've shown you here. So the blood clot forms. Causes of the formation of blood clot is via a series of reactions which activate the next cascade in the next in the cascade. This occurs extrinsically or intrinsically. So what do we mean by blood coagulation happening extrinsically or intrinsically? Intrinsically, this is because the chemicals that are responsible for blood coagulation, they are coming from inside, and the intrinsic is happening because this particular blood is in contact with the foreign substance. Okay, so that's what we mean by extrinsic. So now let's look at extrinsic. Extrinsic clotting mechanism is going to be triggered when blood contacts damaged blood vessel walls or tissues. Then chemical outside of blood vessel triggers blood coagulation. So under intrinsic clotting mechanism, what triggers it first is what we call thromboplastin. Always take note of this. Thromboplastin is found in the blood. So thromboplastin is factor number 3. Thromboplastin is what activates the extrinsic clotting mechanism. So there are a number of events that occurs. When you look at factor 7, factor 10, factor 5, factor 4, as well as factor 2, prothrombin. So now, how is with the intrinsic clotting mechanism? It's going to be triggered when blood contacts a foreign surface. Chemical inside blood triggers blood coagulation. Because chemicals are coming from inside blood, this is the reason why we are calling it intrinsic clotting mechanism. It's going to be triggered by Hegman factor. We call it factor number 12. It found inside the blood. So questions are going to arise: what activates intrinsic clotting mechanism? Intrinsic clotting mechanism is activated by factor 12, which is the Hegman factor. Then what happens is that factor 12 activates factor 11. Follow this sequence. Then factor 11 activates factor 9, which joins with factor 8 to activate factor 10. Okay, it's simple. Factor 12 is going to activate factor 9, factor 11, which factor 11 is going to activate factor 9. Then factor 9 is going to join with factor 8 to activate factor 10. This is the cascade here, the extrinsic and intrinsic. The final destination where we want to reach is at factor V, which is factor 5. So whenever there's an intrinsic clotting mechanism, tissue damage is going to release tissue thromboplastin. Whenever there's a tissue damage, it's going to release tissue thromboplastin, which is factor 3, which happens in the presence of calcium ions. Is going to activate factor 7. Then factor 7 in the presence of calcium ion is going to activate with the factor 10. Okay? Then factor 10 is going to combine with factor 5. The same thing is going to happen in intrinsic clotting mechanism. Blood contact foreign surfaces, then Hegman factor is going to be released, then it's going to activate factor 11. Then factor 11 is going to activate factor 9, because this is factor 12, activating factor 11, then activating factor 9, then factor 9 combining with factor 8, okay, to activate factor 10. Once factor 10 is activated, then they are going to combine with factor 5. This is going to be when we factor X and factor V, we call it a prothrombin activator. Okay, so the prothrombin activator converts prothrombin, which is factor 2, to thrombin. Then this factor 2 converts fibrinogen into a fibrin. Then this fibrin is going to combine with the factor 8, which is factor 13, sorry. It's going to combine with factor 13 to stabilize this fibrin. This fibrin is the clot that you see, that dark thing you see on the wound. Okay? So basically, after the clot has been made, it needs to be what? It needs to be removed so that the healing can be complete. After a blood clot forms, it retracts and pulls the edges of the broken blood vessel together while squeezing the fluid serum from the clot. Then platelet-derived growth factor stimulates smooth muscle cells, fibroblasts, to repair damaged blood vessel walls. They all need now to be repaired so that the skin can be formed on top. Then the plasmin is an enzyme that digests the blood clot that has been formed on top of the wound. And these are some of the clinical correlation. We have hemophilia A. So sometimes people have clotting deficiency, like they have clotting disorders. Some people breathe for a long period of time. They breathe for a longer time than normal. In cases of hemophilia A, in hemophilia A, specifically the question is going to require you to say what causes hemophilia A. Hemophilia A is caused due to deficiency of factor 8. You understood the importance of factor 8 because factor 10 was combining with factor 10 was combining with the factor 8 so that they can form factor 5. So this is the most common infection, the common white blood cell disorders in account to about 85% of all cases, okay? Almost exclusively in males, mostly. Females are usually carriers caused by a gene mutation, okay? So in female are usually carriers, they are caused by a gene mutation due to when the X chromosome is deficient. Okay, so it occurs about 1 over 10,000 male births. Other hemophilias account for another 15%. We have hemophilia B, which happens due to factor 9, deficiency of factor 9. Then we have hemophilia C, that happens due to deficiency to factor 11. Then we also have hemophilia D, which happens due to factor 12, which is the Eggman factor. But the most common one is the hemophilia A, which is caused due to deficiency of factor 8. Okay, we also have thrombocytopenia, abnormally low levels of platelets, usually below 50,000 per microliters of blood. Then we also have thrombus, abnormal clot that develop in a blood vessel. Okay, then we also have embolus, free thrombic clots carries in the blood that usually get caught in arterioles in the brain, kidney, and lungs. Embolus can even cause stroke. Okay, so now this is the last part that we're going to discuss in this session. We're just looking at the practical hematological test for bleeding. When you were to be cut right now, in case you are cutting vegetables and you cut yourself, how long is the bleeding time going to take? How long is the clotting time going to take? And how long is the prothrombin time going to take? So now let's look at the bleeding time. What is bleeding time? This is the time interval that takes between start of bleeding until arrest of bleeding of blood. From someone when blood start coming out until blood stops, how long should it take? Normal duration should be 3 to 6 minutes because the clotting mechanism has been activated and it needs to close up this particular vessel that has been broken. Above this, then you probably have issues with the platelets. Then how about clotting time? What is clotting time? This is the duration of time it takes for blood to clot. Normal duration is about 3 to 8 minutes. Then we have prothrombin time, deals with the duration of formation of prothrombin after addition of oxalate and calcium ions to the blood. Okay, this one needs to be it can take some far away minutes, but it should not take days. Okay, so basically we are going to end here, and this marks the end of blood physiology part 2. And if you've reached this far watching this particular tutorial, it means that it was beneficial to you. Don't forget to subscribe so that you don't miss content that is going to be sent in the future.